Hello everyone, today we are going to discuss about gene transfer technologies. The introduction of new DNA into an existing organism cell using by vectors such as plasmids and modified viruses is known as gene transfer. Gene transfer technologies were originally developed as a research tool for investigating gene expression and function. So the cell that receives the foreign DNA is called the transformed cell, and the process is known as transformation. Transformation is the process of genetic modification in which the fragments of DNA from one cell enter and get integrated with the DNA of another cell. So there are various methods of gene transfer. They are the natural method and artificial methods. Natural agents such as living cells, bacteria, other DNA molecules and viruses are utilized to transfer genes in this natural method. And in artificial method, the exogenous gene is transferred by using artificial means like physical or chemical agents. And they are natural methods include conjugation, bacterial transformation, transduction, agrobacterium mediated transfer. And the artificial methods include microinjection, biolistics or gene gun or particle bombardment method, and calcium phosphate method, liposome mediated transfer and electroporation. So let us see about conjugation. It is a microbiological recombination mechanism that occurs naturally in organisms. So the two living bacteria, a donor and a recipient, come together during conjugation. They connect through cytoplasmic bridges and transmit single-stranded DNA from a donor to the recipient. The additional DNA may either integrate with the chromosome or stay free inside the receiving cell. So the DNA gets integrated or stay free inside the receiving recipient cell. So the examples Salmonella and Shigella cells. The conjugation can happen between these bacterium cells. This is in contrast to the change that occurs within a bacterial genus. As a result of conjugation, genes from two distinct and unrelated bacteria can be transferred. So it is a kind of gene transfer. Transformation. The process of introducing foreign DNA into bacterial cells is known as transformation. E. coli take up plasmid DNA and ice cold calcium chloride and then undergoes a heat shock for around 90 seconds. By this method, the transformation frequency, which refers to the fraction of cell population that can be transferred, is reasonably good. For example, approximately 1 cell for 1000 cells. DNA uptake is stimulated by a short heat shock and a rise in temperature from 5 degrees Celsius to 40 degrees Celsius. Larger DNAs are less effective at transforming in general. Transduction. The transduction process involves inserting genes into the genome of a host cell utilizing viruses. Example, bacteriophage. So bacteriophage is a virus that affects bacteria, and the virus is used in gene transfer because of the following characters. Virus's ability to transfer their nucleic acid into cells, replication and gene expression at a higher level. So to enter the host cell, the foreign gene is packed into viral particles. A receptor-mediated mechanism allows virus particles bearing candidate gene sequences to enter the cell and then into the nuclear genome. The vector genome passes through a series of complicated steps that results in the desired DNA, which can neither remain as an episome or integrated with the host genome, and then the candidate gene is expressed through gene expression. Agrobacterium mediated gene transfer. Agrobacterium tumefaciens is a soil phytopathogen that naturally infects plant wound sites and causes ground gall disease through delivery of transferred tDNA from bacterial cells into host cells through a bacterial type 4 secretion system T4SS. So this bacteria has become the most popular plant transformation tool to date. Any gene of interest can now easily be used to replace the oncogenes in the tDNA region of various types of binary vectors to perform plant genetic transformation with these Agrobacterium tumefaciens. These Agrobacterium tumefaciens mediated plant genetic transformation process requires the presence of two genetic components located on the bacterial TA plasmid. And the first essential component is the tDNA, defined by the conserved 25 base pair imperfect repeats at the ends of the tDNA region called border sequences. Second is the virulence region, which is composed of at least 7 major loci, VAR, A, B, C, D, E, F and G, encoding components of the bacterial protein machinery mediating tDNA processing and transfer. The steps involved in Agrobacterium tumefaciens mediated plant transformation process are attachment of Agrobacterium tumefaciens to the plant cells, sensing plant signals by Agrobacterium tumefaciens, and regulation of virulence genes in bacteria through transduction of the sensed signals, generation and transport of tDNA and virulence proteins from the bacterial cells into plant cells, nuclear import of tDNA and effector proteins in the plant cells, tDNA integration and expression in the plant genome. So you can see in the diagram, the Agrobacterium tumefaciens infects the plant cell and delivers its components into the cell. Thus the gene is transferred, and the desired DNA can be transferred by using this method. It's bacterial plasmid. And the next method is the microinjection. Microinjection refers to the injection of DNAs or cell organelles directly into cells using a microinjection needle. By this method, RNAs, DNAs, proteins or cell organelles are injected into animal cell zygotes and plant protoplast. For microinjection, a stereoscopic dissecting microscope, a micropipette and a glass injection needle are required. For cultivated cells, DNA transfer through microinjection is commonly employed. By this method, it can be used to deliver DNA into big cells like oocytes, eggs and early embryonic cells. And it is a method of transfection, refers to the chemical or physical transfer of DNA into eukaryotic cells. And the micropipette tip is a diameter of 0.5 mm is used to transfer foreign DNA under a powerful microscope. Microinjected cells are put in a container. A holding pipette is placed in the microscope field of vision. Sucking and holding pipette and holding target cell at the tip. So you can see in the diagram, my holding pipette is used to hold the cell, and the injection needle containing the DNA solution is injected into the cell. Thus the gene is transferred. So transfer the contents of the needle into the cytoplasm. Biolistics or gene gun method. It is also known as shooting the plant or animal cell by DNA coated gold or tungsten particles. And the instrument used to shoot the DNA into the cells is called gene gun. It is a physical method of DNA introduction. In this method, no vector is used to introduce DNA into the cells. Gene gun consists of gas acceleration tube, a firing pin, a macro projectile and stopping screen and target cells. The DNA is mixed with tungsten or gold particles of microscopic size and is treated with calcium chloride or PEG. Biolistics. And this method, the microprojectiles are positioned on a holder about 5 cm above the host cells within the chamber of the gun. and in this chamber is sealed with helium gas so that it can pumped into a small compartment above the gold particles. The microprojectiles are fired into the cells with a speed of 300 to 600 meter per second. And next method is the calcium phosphate method. It is a chemical method. Calcium phosphate is used as a carrier of rDNA. And the steps involved are DNA is dissolved in a phosphate buffer and the calcium chloride solution is added. So calcium phosphate combines with DNA to form calcium phosphate DNA precipitate, and a precipitate is added to the host cells. And thus the precipitate particles adheres with the cell surface and the cell engulfs the particle with DNA by phagocytosis. That is, the engulfing of these particles by DNA is called with the DNA is called phagocytosis. And the DNA enters the cell and reaches the nucleus and gets integrated. And next method is the liposome mediated gene transfer. Lipofaction is the process of introducing the DNA into host cells through liposomes. Liposomes are vesicles formed of phospholipid bilayer, and it resembles the plasma membrane, so it readily fuses with the plasma membrane and delivers the DNA into host cell. Electroporation is a process of introducing DNA into host cells by increasing the permeability of the cell by applying an electric field. It is a physical and chemical method of gene transfer. The instrument producing electric field is called electroporator. So the setup consists of cells in suspension in a cuvette and the two electrodes, cathode and anode, and power supply is given. So it increases the permeability of the cell membrane and the DNA is introduced into the cells. These are the various methods of gene transfer. Thank you.